So it is a great honor for me to introduce the first keynote of this 2023 edition of JOBIM, and Dr. Allison Balandras-Colas. So Dr. Balandras-Colas obtained her PhD in structural virology at the University of Lyon, and then she made a first experience as a postdoc at the Dana-Farber Cancer Institute in Harvard Medical School. And then she also went to the Francis Crick Institute in London, UK, where she specialized in cryo-electron microscopy. Now, Dr. Balandras is a permanent researcher in CNRS at IBS Grenoble. Donc, without any further ado, I leave Dr. Balandras.
Thank you very much. Thank you for the kind introduction. Maybe you can just help me to set up my presentation, please. No, now it's all. Yeah.
So you can also show.
Yeah, but now it's all fine with it's all inserted. So it's whatever, it's fine. And maybe just.
I think that's fine.
Okay. All right. Well, thank you very much for this invitation. It's really a great honor to present my work today. As Eduardo was saying, I'm a social biologist and I focus on virology. So I take a special interest into the retroviruses and to see how they function at the molecular level. So I'll give you, that's the outline, a brief introduction, and then focus on the intesome complex from the beta retroviruses, then switch to the lentiviruses, quick stop with the cellular partners and inhibitors of integrase. All right, so retroviruses are RNA virus, which retrotranscribe the DNA, the RNA genome into double-strand DNA, and then inserted into the host cell genome. They are classified into 7 genus, and the most known one is the lentivirus genus with HIV-1, so for human immunodeficiency virus type 1, which is responsible for AIDS, for the syndrome of AIDS. But that's not the only retrovirus, and although there are 7 classes, genus, they all together have the same life cycle, well, roughly. So this is a very brief description of the life cycle. So they have an envelope protein at the surface of the virus that recognize a receptor at the surface of the membrane of the cell, which triggers the entry of the capsid, release the RNA genome, which is retrotranscribed by the retrotranscriptase into double-strand DNA. And that DNA is imported into the nucleus, where the integrase protein inserts this viral DNA into the chromosome. Let me get the pointer. No, here. Yes, okay. So yeah, at this stage, the viral DNA is inserted into the host chromosome by the viral protein integrase. And after that, the late stage of the life cycle occurs. So transcription, replication of the virus, genome encapsulation, and then protege virus are being exported by budding. But so this step of integration is actually the point of no return. That means once the genome of the DNA is inserted into the host cell, there's no coming back. You are infected for life, and although you may not develop the AIDS, you you have the virus DNA in your cells. So that's a very key step for the life cycle. So how does it work? We know that the integrase protein multimerizes at the end of a viral DNA, and it will actually cleave 2 nucleotides at the 3' end of a viral DNA. Then it anchors onto the host DNA chromosome, where it will nick the target DNA and insert the 3' end into the host chromosome. And after that, so the DNA of the virus is actually called a provirus. It's inserted into the chromosome, and the insertion is actually on 2 opposite strains of the DNA, the double-strand target DNA. And so cellular protein will actually fill the gaps and repair the DNA. So all retroviral integrase share the same topology. So they have 3 domains: the N-terminal domain, the core catalytic domain, and the C-terminal domain. They all fold into the same secondary structures. And although the sequence is not highly conserved, it's about 15 to 20% of identity between different retroviral integrases, they all fold into the same secondary structure of the organization. And these domains are actually linked by flexible linkers. This is why very early on we got the structure of individual domains, either by crystallography or NMR. But to get the structure of a full protein, that it actually became a huge challenge. Also, so HIV-1 integrase was discovered in the 70s, late 70s. And people have tried or scientists have tried for a very long time to get the structure of that protein, but with no success. And it happens that this protein, HIV-1 integrase, is actually very insoluble and tends to aggregate in vitro. So quite a tricky protein to work with. It's only in 2010 that Peter Trepanov came up with a brilliant idea. He looked into other retroviral integrase and find out that the one from the spumavirus, the prototype for the spumavirus integrase is actually very soluble and very active in vitro. And he was able to crystallize it in complex with 2 oligomers, synthetic oligomers of viral DNA that actually mimics the end of the genome of the spumavirus. So let me describe briefly this structure to help you picture how it works. So this is the same structure. It's called an intazome. That's the complex formed of the integrase plus the viral DNA. And so on one end, we have a dimer of integrase here, a second dimer here in green. In the first dimer, we see the inner protomer in red. The full-length protein structure is actually resolved. But the second protomer, the outer one, we only see the catalytic core domain. We don't see the N-terminal and C-terminal domain. We don't actually know where they fall into the structure. And what's interesting is that the catalytic core domain of the inner protomer here engage the end of the viral DNA with the 3' end in the active sites here. And the N-terminal domain actually go to the opposite strand of DNA, and it holds the opposite strand here. The catalytic core domain acts as a bridge between the 2 dimer of integrase, and it holds the complex together. So the inner protomer in this extended form, symmetric to the second dimer in this extended form as well. So we thought, okay, that's probably how it works. They have the same folding, so most likely all retroviral integrase fall into the same architecture. But we wanted to make sure of that. And this is where I start my first postdoc in Alan Engelman's lab. This is a virology lab, and they gave me this quite risky project trying to get a new intazome structure from another retrovirus. So we knew for sure not to start with HIV-1. Scientists have tried for decades to try to get the intesome structure. Well, there's no way I can do better than others. So we focused on other integrases that were not so well or never described in vitro. And for that, we looked into the WDSD DSV integrase from the epsilon retrovirus, RevVe from the gamma retroviruses, LPDV from the alpha retroviruses, and GSRV and MMTV from the beta retroviruses, knowing that PFV here is the one that's known. And it turns out that indeed that was a challenging project, as these proteins in vitro are either insoluble, or I could not get enough material to work with, or more insoluble, or would precipitate along the purification process. It's only MMTV that gave me something. I was able to solubilize the protein, purify it, and get just enough material to work with. And also, I also checked that the protein was active in vitro, because it's no good to have a protein soluble if it's actually not active. So I spent quite some time to find a protocol where I could assemble the complex of the MMTV integrase with viral DNA ends. So same trick. We used short oligomers that mimic the sequence of MMTV genome. And well, after lots of trials and errors, I was able to assemble this complex here that elutes first on the size exclusion colon. Here is the integrase being eluted by itself and the viral DNA by itself even later. And so I characterized this complex. The first thing was to make sure that it's actually an active complex and not just some aggregates that, well, binds together somehow. So for that, we used a strand transfer activity assay. So this is the schematic of what happens, and it's used very broadly in the field. So we mix either the intesome or integrase and viral DNA not pre-assembled with a target plasmid. And what we can expect is the insertion of 2 viral DNA ends on that plasmid, which after deproteination should yield this linearized plasmid. One side product is the half-site integration, where only 1 viral DNA is inserted onto the plasmid, and that means it's a dysfunctional complex, because the complex should hold the 2 ends of the genome. And that's the type of gel we can actually get. So that will be the plasmid with no insertion, with integrase and viral DNA. So viral DNA will migrate here. We see now a band of linearized plasmid. So that's the product of concerted integration, meaning that our protein is actually active in solution. And when we compare to the intesome, it also yields the same product of integration and even a smear. I don't know if it's, well, it's barely seeable, but there's a smear here, meaning that there's multiple integration into the linearized product that yields even smaller pieces of DNA. And interestingly, with the intesome alone, this is where the band where half-site integration may occur, and there's nothing in the intesome, but there is some a band of higher intensity for the integrase alone compared to the plasmid itself. Okay. So we have an active complex. We've done lots of characterization to try and understand how that complex behave, biochemistry. And I've tried for, well, a few months to get the crystal structure, thinking that if I follow the lead of PFV, they were able to crystallize it. There's no reason why the MMTV intesome structure would not occur by crystallization as well. But somehow that did not happen. So we had to call for a collaboration with Dimitri Liamkis at the Salk Institute, who's doing cryo-EM, cryo-electron microscopy. At that time, that was a very unknown, structural biology technique, or not so well used. But still, he got back to us within a couple of months with this map, with this EM map. And that's where I started to think, okay, I need to look into this technique that looks quite interesting. So he got us this map. It's overall at 6 angstrom resolution. In the core density, it's a bit higher resolution, meaning that the side density is less resolved, so probably a bit of movement. And in the meantime, I had worked to get the individual domain structure of MMTV integrase, and I was able to fit them into the map. And to our surprise, we could actually fit 8 catalytic codomain, 8 N-terminal domain, and 8 C-terminal domain, as well as the 2 viral DNA ends. So unlike PFV, there's actually 8 integrases forming this complex, and that was unexpected. So we looked into, well, so first of all, we connected all of the domains. We built the linkers in between the domains, and there were no doubt about how or where the linker would go and the pathway. And we compared the 2 intesome structure. So they're actually not so different. They have the same organization in the core part of the intesome. So they both have the 2 viral DNA ends, that's expected. And they have the core catalytic domain in red and green and red, engaging the 3' end of the viral DNA. And one catalytic codomain donates its N-terminal domain to engage or embrace the opposite strain of viral DNA. And it holds the second half of the intesome together. Now, the main difference comes from the C-terminal domain that are here positioned at the synaptic position in between the 2 halves of the complex. So in the case of PFV intesome, this C-terminal domain is provided by the inner protomer of integrase, while for MMTV, this domain is provided by the flanking dimer. And we thought, well, this is quite strange. How come MMTV could not just use, there's actually 8 C-terminal domain. How come it does not come from the catalytic integrase subunit? So we looked at the linker between the catalytic core domain and the C-terminal domain. So this is a side view of PFV integrase, the green one here. And the linker between the catalytic core and the C-terminal domain is actually 50 amino acid long. Whereas in MMTV integrase, this same linker is only 8 amino acid long. So that most likely is the reason why it can't use the C-terminal domain of the catalytic subunits to occupy this space here, and it needs an extra integrase to bring this C-terminal domain. It happens that MMTV integrase and solution is dimeric naturally, and so it's not just one integrase that comes filling in the gap, but a dimer. So there's an extra dimer just to provide one domain. And so using this linker length, we could extrapolate about the rest of the intesomes. And we realized that the gamma, epsilon, and spumavirus genus, they all have a very long linker. So we can expect that most likely they all resume a tetrameric architecture. While for the beta and alpha retroviruses, they have very short linkers, so most likely they will all assemble into an octamer intesome. As for the lentivirus and delta retroviruses, they have this intermediate length, so we can't really tell. They could do either way. And while we're doing this study, our competitors came up with the crystal structure of the RSV intesome. So it is doable to crystallize it. I just didn't manage. And it indeed assumed this octameric architecture as we predicted. So we actually co-published this data in the same journal. And I guess it worked in our way that we had two different intesome structure that both conclude the same octameric assembly. So by the end of this postdoc, many questions arises. So first of all, what happens for the lentiviruses and the delta retroviruses? Are they going into a tetramer or octameric or something even different? But also, what about this cool technique cryo-EM that looks quite promising? So I joined Peter Cherepanov lab in London, and he was already looking into integrase from the lentiviral genus looking for a candidate that would be soluble, because HIV, we couldn't manage to get it soluble and to form the intazome. And he used the Medivisna virus integrase, the Visna integrase, to actually be the perfect candidate. So it is more soluble than HIV-1, and he was able to purify it and to have it active in solution. So when I joined the lab, we were already at the stage where we found a protocol to assemble the intazome complex, and this is what it looks like. So it actually elutes very early on a superdex, on a size exclusion colon, meaning it's probably a very big, either it's an aggregate of protein into DNA, which is very likely. That's exactly what HIV does. It just aggregates, soluble aggregates. So either it's a soluble aggregate or it is actually a very big complex. And when we tested the activity, this fraction here is actually very highly active. So we have a strong band here and a big smear as well. And when we use dolutegravir, that's a known inhibitor for HIV-1 integrase, it actually completely abolishes the catalytic activity of this intazome. So it looks like we have actually a complex that's active, but it's not enough, because we know HIV-1 integrase is able to continue its catalytic activity even when it's aggregated. So we further characterized it by looking at the sequence between of the junction between the viral DNA and the target plasmid. So that's a common, well, not so common, but it's widely used now to see where retroviruses integrate their genome, and for that they're using, well, next generation sequencing. Here we used linker-mediated PCR and sequenced these data on an Illumina platform, and this is what we got. So we compared the product of integration from purified Visna intazome with plasmid target, with a target plasmid, or purified intazome with genomic DNA from sheep cells. That's the actual natural host for the Medivisna virus. And compared to sheep cells directly infected with the Visna virus. And in all of these cases, there's a gap of 6 base pair between the 2 sites of integration. So we know they happen on opposite strand of the target DNA. So there's a gap of 6 base pair, but also the nucleotides before and after the site of integration are conserved in all of these experiments. So meaning that the purified intazome actually behaves the same as the virus in the host cells. So with that, we're actually quite confident that this, whatever comes off very early on the colon, is an active complex and not just aggregates. So that's why I wanted to learn about cryo-electron microscopy. So you may have heard that the technique was awarded the Nobel Prize in 2017. And this comes from major work on how to prepare the sample, the software acquisition, beta detectors, and the processing of the images. So I want to give you a very brief overview of how this technique works, because it's everywhere now. So, well, and also, yeah, so it's getting better and better. So within just the span of 7 years, we can now reach up to 1.7 angstrom resolution from just 80,000 particles. So that's really nothing. And the current best resolution is 1.22 angstrom from Shaw's Sherris lab, the guy who developed the one of the main processing pipeline. And although these are using proteins that very well behaved in this technique, it is very impressive, because about 10 years ago, cryo-EM was called blobology because we could not get higher to 10 to 15 angstrom resolution. So what about the pipeline? How does that work? So very briefly, but just again to give you an idea about the amount of work and energy you need to put into this flow to get a high resolution structure, which is not a given. So first of all, one should spend a lot of time on protein preparation. Whatever you will put on a microscope, you will see. So if you put crap or aggregate protein or contaminated stuff, you will see it and you will make your life very hard to actually distinguish between what you want to study and what is actually on your grid. So spend a lot of time at the bench, very important. Then check your sample by negative staining. You can't reach very high resolution with this technique. It's really just covering your sample with heavy metal, but it gives you very quickly an idea about how well your protein behaves. Is it homogeneous? Do you have a protein that's pure, but maybe you have dimer, tetramers, aggregates, and it's very heterogeneous, and then it is hard then to analyze. And once you actually have this step optimized, then you can move to cryo. I mean, here the contrast is very well. You can really see the proteins, the white on gray background. When you switch to cryo, this is the type of contrast you have, and that's a good contrast. So it's very hard to see what you have. So check by negative staining, that way you know exactly what you can look for in cryo conditions. And then in cryo conditions, you need to screen, optimize the way you're screening your samples, because of course it's protein dependent. So you may want to go through quite a lot of freezing to find the best solution. Do you need detergent? Do you need a very fast, short blotting time? Do you need low salt, no glycerol? That's a given. And even then, it's not always easy to see your particles. So this is just an example of things that can go wrong. So here, so that's the edge of the grid. So we want to have the grid, the protein in these holes. So very small, thin ice. And what you see at the top is the edge of the support. So this you can't collect on. And this is where you want to have your samples. And you can actually leave a protein here, just at the edge, at the rim of the grid hole. Most likely it's all aggregated on the carbon support and it's not entering into the hole. So you also have to convince your protein to go in the holes. So here, not usable. Here we have protein aggregated. So very heterogeneous samples. There are a few isolated particles, but most of it is aggregated. So, well, it is in theory usable. I mean, you can actually select for individual particles, but it's going to be a pain. It's probably better and easier to go back a step and try to improve your sample. And in here, we have liquid ethane trapped into the vitreous ice. So contamination with your freezing ethane. So that's the kind of grid or samples you want to see. So there's a nice distribution, and yes, we don't see much. So that's why you need to do the negative staining first to know what you're looking for. But there's not a lot of contrast, and that's expected because you want something very, well, not a lot of electron, such that you don't damage your protein. You can have higher contrast if you improve the amount of electron, but then you can't get to high resolution because your protein is all damaged by the electrons. So it's all about compromise. And so here we can actually see, so they all are individual particles, not aggregated. That's a good sample. I just didn't pick all of them. So once you have a good sample, you can move to collecting on a high-end microscope. So most of the time, people go on Titan Cryos and then do the pipeline of analyzers. So it's a long journey. First, you have to collect the images. So around a couple of thousands movies. That's usually a good start. So we collect movies. So these are actually exposition of your sample for a few seconds. And we call it movies because instead of having one image during which the particles actually move along within the ice, although it's at -170 degrees, it's in vitreous ice, still the particle moves under the electron beam line. So by collecting a movie, so an image, you don't move, but you collect frames of that same of that exposition, you can then realign all of these frames and have a much better contrast and a much better resolution of your particles. So that's first thing, collect the movies and do the motion correction, align the frames. Then you need to estimate the CTF, the contrast transform function, to actually see if you have signal information at low resolution, well, high frequencies, low frequencies. If you don't get to high frequencies, then you can't go to high resolution. It could be a problem with your ice thickness. It can come from many things. Then you need to pick your particles from each of the 2,000 movies, well, which are now images. Then all of these particles, you need to classify them. So basically to classify them into the same views. So what happens is that your protein is in all orientation trapped in the ice, and you're taking a screenshot from the top. And we want to classify all of the top view, side views, bottom views, and you classify them into these 2D classes. And from that, you can go to the 3D reconstruction. So there's a lot of bioinformatics algorithm. I cannot explain any of them, but it's very, it's been moving very fast in the last 5 years. And actually data that I collected 5 years ago, I could reach about 4 or 5 angstrom resolution. And thanks to the advance into the development of this algorithm, processing the same image, I can now go to 3.5 angstrom resolution. So there's really like, the technique is really moving fast for a while. And then, so usually you do the first ab initio reconstruction at low resolution, and then you do refinements and post-processing, and you can then go to very high resolutions. And so I've learned, well, I used all of that to get the first prion structure of, there you go, of this 9 tasome. So it's massive. It's huge compared to the other tasomes. We can actually see the viral DNA here pointing out. It's very highly defined in the core, less defined in the outer domains. That means there's a bit of movement in the outer part of the structure. That's expected. More DNA here and there. And with that map, at about 3.8 angstrom resolution, we were able to build the model. So we had the crystal structure of individual domains, but it was very easy to dock them into the map and to connect, to build the linkers and to build an atomic model. So that's the final model in the map. So it has the viral DNA ends. So there's one hand here, second hand here, and there's a piece of target DNA here. That was, well, we designed it that way. And then there's 2 catalytic tetramers. So we jumped from a tetrameric intasome with a PFV to an octameric intasome with MMTV, and now it's a hexadecameric, so 16 integrases in this complex, and not just one dimer at the catalytic interface, but a tetramer. Same organization, catalytic core domain, engage the end of the viral DNA and exchange its N-terminal domain to lock the opposite side of the intasome. And there are also flanking tetramers, so just like for MMTV, which donates this one synaptic C-terminal domain. So of course, it's not the same, the only role of this outer tetramer. It actually holds the whole structure together. So there's a big network of interdomain interactions. But so we initially thought that all of the retroviral intasome will assume this architecture with 4 integrases. We were quite wrong. They get bigger and bigger, and that's probably why we couldn't get the intasome crystal structure of HIV-1 in the early days. And yet, they have the same organization very close to the viral DNA ends. So there's always the dimer with the catalytic core domain that engages the end of the viral DNA and the synaptic C-terminal domain, bridging the two half of the complex. And while we were doing this work, our initially collaborators, Dimitri Ryamkis, became my best competitor, very friendly one, but still. And he managed to get the intesome of HIV-1 by cryo-EM. So we have to say that it's a very heterogeneous mixture of particles. So they observed tetramers, dodecamers, hexadecamers, and even bigger aggregates. So it's a very tricky particle to work with, but they managed to get a very nice structure. And it's exactly comparable to the one we got from the Visna virus intesome. The two viral DNA hands here, catalytic tetramers here and there, and flanking tetramers. Here, they don't actually see the last two dimers from that flanking tetramer. And that's because they used a domain fusion protein to solubilize their HIV-1 integrase. And we think that it makes steric clashes. So what about the linkers? Can that explain this big structure? So if we look at all of the, so here I overlaid all 16 CCD to CTD structures, and I got rid of the N-terminal domains. And now the linker is alpha helical. And that's true for all lentiviral integrases. They all have a alpha helix between the catalytic core domain and the C-terminal domain. So we can extrapolate that all of intesome from the lentivirus will actually resume a hexadecameric intesome assembly, or at least a mixture because of this rigid linker. As for the delta retroviruses, so a couple of years later, another team actually solved the crystal structure and the cryo-EM structure of the HTLV1 intesome. And that linker is not forming alpha helixes, but a flexible linker. So it actually falls into the tetrameric architectures. So, okay, that's quite fun to see, well, that this one protein, it has the same function in all of its viruses, but it assumes different architectures. And we're wondering why, it has to have a biological meaning to that. So we looked into the interaction with cellular proteins, host partner. And there's actually one that's well known for the lentiviruses. It's called LEGEF. So LEGEF stands for lens epithelium-derived growth factor protein 75 kilodalton. That's actually very misleading. Well, that's how it was discovered first, but actually it's not in the lens epithelium, but it's found in most tissues. It's not a growth factor, it's actually a transactivator cofactor. And although it does migrate at about 75 kilodalton on a denaturing gel protein, it's actually 60.1 kilodalton, so it's just a protein derived, I guess. What it is really is a chromatin-associated factor, transcription coactivator. It specifically binds all of the lentiviral integrases, and it helps targeting the integration into actively transcribed genes. So we think that's the most, well, it's the most meaningful function of LEGEF in the context of an infection, because obviously it has a role in cells that are not infected, but in the context of an infection, then the intesome interacts with LEGEF, and that targets the integration into actively transcribed genes. And LEGEF is actually made of this IBD domain. So for integrase binding domain, we don't actually know what's the role of this protein outside of an infection, but it's here in regular cells. There's an AT hooks here that binds to DNA, and a PWWP domain that binds to epigenetics marks, trimethylations on the histone-free lysine 36. and that's a mark for genes that are transcribed. So we managed to get the structure of the complex with LEGF, or should I say just the one integrase binding domain. So we use full-length LEGF, but we could only see this domain in the structure. And it attached to the catalytic tetramer. Actually, so it goes into this pocket here. And that's exactly where it was found crystallized in 2009, where the scientists have used just the catalytic core domain with just the IBD domain of LEGF, and it goes into this pocket here. There's actually now inhibitors of this interaction, and they work pretty well, but not used in the antiviral strategy yet. But so the interesting thing is that there's 16 integrases here. So 16 possible sites where IBD could bind and attach on this intesome. So why these two positions? Is this the only place where LEGF can bind? So to answer that question, we set up an experiment. Oh, no, maybe not. Too soon, too hot. Yeah, okay. We set up an experiment with collaborators at the Creek, Hassan and Dominica, where they do single molecule imaging. So what we did is that on a chip, we bound intesome labeled with sci-fi, sci-fi on the viral DNA end. So each dot here is a single intesome attached to a chip. And then we flowed in LEGF with a different label, and we washed out the unbound protein. And now we can see that all of the dots co-localize with the fluorophore for LEGF, especially when we merge them. And what we did is a photo bleaching experiment. So we, using a laser, we photo bleached the chip, and we measure the steps of decreasing intensity of the fluorophore label. And every time it steps down, so every time the signal decreases, that means one molecule of LEGF has been shut off. So the label is not flashing light anymore. So we could count these steps, and that's what it looks like. And each steps correspond to one molecule of LEGF attached to this one intesome. And we did statistics in different buffer concentrations. It turns out that at near physiological buffer, there's on average 6 molecules of integrase bound on one intesome. And even at very high concentration of salt, there's still 2 molecules of LEGF attached to the intesome. So we use low salt buffer in our experiment to actually get this structure. So how come we see only 2 molecules of LEGF? So that was a puzzle, and we think that it's because these 2 positions are the most preferred one because they're close to the target DNA, and it makes more sense to be here. But there is actually room for a lot more LEGFs that just spread out in a non-even way. And so with the merging of the data and the way the data is processed for cryo-EM, then we lost this information on the way. So we think it acts as a sensor platform for getting into the chromatin. And we wanted to see if, because we know LEGF is actually sensitive to epigenetic marks on the histones, if it would direct the integration into nucleosomes with this mark or without this mark. So we tried to do that in vitro. That was quite an interesting problem to tackle. So first we had to make this polynucleosome array. So that's a repetition of 12 nucleosomes with or without the epigenetic mark on histone 3. And this is actually, we checked by a negative staining that the polynucleosome actually looks like what we want it to. So we can really see each ball or each ball for the nucleosomes, and we can even see the DNA linking them. And so we use that as a target DNA for our intesome activity. And it works. Here we have a gel post-integration, and we can see the different projects of integration. And they have this very laddery pattern because they don't integrate randomly along the polynucleosome, but they have preferred sites for integration. And I tried to compare integration or to make a competition using a polynucleosome that was labeled with one label with no trimethylation on histone 3 versus a polynucleosome with a methylation. And using that technique, so I have very pretty gels, but I could not actually detect any preferred integration into one of the other target DNA. And that we think is because although it's a very nice system, it's an in vitro simplified system where the chromatin is not compacted and uncompacted. And we think what happens in the cell is that LEGF actually carries the intosome into the actively transcribed genes that are open chromatin. And then the intosome can insert the viral DNA in these regions. And in our system, the polynucleosome without the epigenetic mark actually is not compacted. So it probably is what makes this competition null. Okay, last part, inhibition. So there are inhibitors of integrase that are used for the treatment of patients infected with HIV-1. They are part of the antiviral therapy, and they have now a second generation, very, very potent drugs. We wanted to see if we could visualize them into the nucleosome, into the intesome, and see how the inhibition works. For that, we still don't want to work with HIV-1, but we have this model using the simian immunodeficiency virus of this macaque. So SIV RCM integrase that is very close to the HIV-1 integrase. It has about 75% identity, which is a lot better than HIV-2 compared to HIV-1 that has only 66% of identity. And so my colleague managed to get the intesome structure of, or to get the intesome solution of the SIV. That's the peak here, and it's active. She also managed to have some formation of intesome for HIV-1, but very little compared to SIV, and it is active as well. So we decided to keep going with the SIV-1, SIV RCM, and so that's the type of particles we have. So this is negative staining, so we see clearly the particles, and we can easily see that it's very heterogeneous. But there are very small particles and these very long stacks. But if we look closely, they are actually all, so this is purified integrase, and they are very repeatedly, like it's very ordered. It's not an aggregate, a regular aggregate. So we thought, okay, if we do 2D classes, we can actually find intesomes that are hexadecameric, that's expected. Some that are missing a dimer here, so only 14 integrases. Some that are missing a full tetramer, or 2 dimers here and there, or even, well, 6 integrase here. And we have these long stacks. And this is MVV, the Visna virus intesome for comparison. So that looks exactly the same. So now that you know a little bit more of the intesome, you can recognize this shape here that we can find here, there, there, there. These are the catalytic tetramers of integrase binding the 2 viral DNA ends. And we actually see them very clearly in the stacks here. So we decided, okay, we're going to take advantage of this loophole to get to the structure of the core center of the intesome. Although it is not biologically relevant, but it's a great tool. And indeed, we were able to solve the structure of these stacks to very high resolution. And if we take only these 4 tetramers, we recapitulate this 9 tasome. But what happens is that 2 tetramers of the same color engage 2 viral DNA ends, and they donate 1 C-terminal domain to the neighboring 2 tetramers. So it forms these stacks. And we know this is not biological because all of them, each pair of tetramers holds 2 strains of viral DNA. So there's a repetition of viral DNA ends that cannot happen in the virus because there's only 1 genomic virus. There's only 1 genomic DNA of the virus, so only 2 ends. So these stacks cannot happen during an infection, but for us, it works because with this repetition, we have more signal, and so we could get to very high resolution structure, up to 2.6 angstrom in the core. It resumes the same architecture. I'm not going to go back on that. Now you know it. And we could actually see, so this is the active site without a drug. This is with dolutegravir and with bictegravir. So we could see very highly and very nicely the inhibitors into the active sites. And this is what it looks like. So it actually comes close to the end of the viral DNA. This part here actually stacks with the neighboring base. These 3 oxygen here coordinate the 2 metallic ion in the active sites. And this larger part here actually occupies the space in the active site. So it mimics a base. And of course, this cannot be integrated into the target DNA. So it stops the integration and it has a very long life cycle. And we can actually also see that one of the resistance that appears is the mutation of this glutamine into a histidine. And this glutamine coordinates a molecule of water here, but then coordinates the metal and the 3 oxygens. And with the histidine, we can't bind to this molecule of water, and so the inhibitor does not stay into the pocket. So with that, I'd like to conclude that over the span of these 15 years of work on integrase, we now know that there's a large spectrum of different architecture of intesomes, although they do have the same function. And we think that the different size corresponds to the ability to bind to cofactors. We also know more about how the drug binds into this intesome and how to maybe analyze and create better drugs. And the future, I think, for this work would be to visualize the intesome in cellulo and try to see what happens in the cellulo. I just hope that next year I can present you the work I'm doing now at the IBS. So we're working on another virus, influenza mostly, and the Borna virus. So yeah, next year, hopefully I can present a different type of work. And I would like to thank all of the people that have been involved. I know it's already time, so thank you very much for your attention. I'm happy to take questions.
Thank you so much, Dr. Balandras-Colas, for this fantastic talk. So we haven't received yet a lot of questions online. I remind you that it's, still possible to do it. There is actually only one, but actually I would like to prioritize if there is any question from Nice. The public here. Okay, then in this case, Ludo Mallet is actually asking, un mode semble pouvoir faire des plus petits fragments que les modes 3. I think it was in the at the beginning of the section about inhibition, right?
So can you read the question again, please?
Un mode semble pouvoir faire des plus petits fragments que les modes 3.
Yeah,
Voilà.
with the polynucleosome. It's not too far.
Yes,
Right,
exactly there.
here, this. So
Yes.
that's actually the bottom of a gel. So it's most likely viral DNA that hits the end of a gel or just from some reflection. So I don't think that really matters. But indeed, we don't see much on the screen here. I don't know what happens online, but there are smears all along. The difficulty with that experiment is that you have to measure the intensity of the fluorophore for each band, not this one, because this is not integrated, but all of the smear. And that's actually quite tricky to quantify. We tried different method, but yeah, I couldn't see a preference for the fry methylated versus unmodified nucleosomes.
Thank you. I actually had a question myself. So, but it's a general one. So lentiviruses are also used, if I'm correct, for gene transfection in gene therapy, right?
Yes, that's right.
So I would have thought that the integrase had some kind of a high specificity for binding to very specific locations in the DNA. You have, of course, in your talk, I mean, you've talked a lot about LEGF, etc., but it looks like it's not as specific as.
Yeah, indeed, it's not. It is not random, but it's not as specific as CRISPR-Cas9, for example. It is still like a broad, a general direction. So each virus has this specificity between the different genus. Indeed, the lentiviruses go preferentially into active genes, but it looks like one cofactor CPSF6 brings it toward a general area of the nucleus, and then LEGF brings it toward a more specific regions. But then there's an even more locally integrase will actually read the DNA and find a specific a sequence that pleases the complex better, but it's not so specific, and so it's not. It was quite hopeful in gene therapy, but I'm not sure it's actually going very well. I don't know if they developed it anymore. It is used in the lab when you want to do in vitro or modification of your cells to create a new cell line. You can use lentiviral tools to do that. But yeah, I don't think it's actually as promising as it was in the beginning for gene therapy.
Thank you so much. Let's see. Is there any question from the audience here? Yes. 1 second, though. I'm sorry.
Can you hear me?
Yeah.
Yes. I don't know if the people outside.
I'll repeat the question.
Okay, thank you. It's thank you very much for your nice talk. It's naive questions. It just do you think we can apply the same kind of approach for transposable elements, not the highly related to retrovirus?
Yeah, there is actually work in Marseille on a transposase, and yes, there is work. It is very close. So they have, at the domain level, they have a lot of similarities. In the way it assembles as a complex, it's not so conserved, but indeed they are very similar in activity and in the way they are specifically interacting with DNA.
Okay, thank you.
Any other question? Is there anybody online? Okay. Then in this case, let's thank again Dr. Balandras-Colas.
Thank you for inviting.